Here we go. Okay, so thank you very much for the invitation. I'm going to give you a tour de force through one of the other major C&M genes, which is titin. And titin is one of these proteins that keeps the motors together, the motors of our muscles. And if you think of a motor like this rather lovely BMW V8 engine, German-made, of course, if you want to repair a motor, you take it out of the car and you dismantle it, and you have all these parts. And if you are a good car mechanic, you know how they fit back together again, and you can identify the defective parts. But in muscle, of course, that isn't possible. We can't take them out. We can't repair them in a garage. And quite often, actually, we are baffled by a picture that looks very much like this does to me. I don't understand what most of these parts are, and I wouldn't be able to put it back together again. And in muscle, we have the same problem. Muscles are machines made from proteins, and they are extremely intricate assemblies, extremely regular. So as you see in this picture here, this is a light microscope image of a skeletal muscle, and you can see that it is beautiful and very regular, and proteins which are labeled with glow-in-the-dark molecules here are arranged in superb order. And if we look at even higher magnification using an electron microscope, you can see these filamentous structures. They're called myofilaments, and some of them are made from myosin and actin proteins mentioned already. But what you can see is that all of these molecules know exactly where to go in this structure, and they do this repetitively from here to here to here. It repeats and repeats and repeats. So how do these proteins know where to go? And the protein that Heinz mentioned already, titin, seems to be the one that is responsible for telling hundreds and millions of other proteins with regular precision where to assemble in this structure. So titin is the one protein that goes from one end of the sarcomere here at this structure called the Z disc all the way to the M band. So it's the only protein that is big enough to span this repeating unit as a single protein. So it's really, really big. But what is big really? So if you think of a person here being a sort of small molecule and an Airbus 380 being something like myosin, then titin is the titanic thing that is really very much bigger than any of those. And just to illustrate that, in an actual picture of the molecule in the same scale as a sarcomere, this is a sarcomere in an electron microscope with myosin filaments, actin filaments, and these are isolated titin molecules, and they're just absolutely gigantic. Each molecule is over 1 micrometer in length, so you could see it in a light microscope if it wouldn't be so extraordinarily slender, so that is just too thin. But it's an absolutely gigantic protein. It's encoded by an absolutely gigantic gene with over 300 kilo base pairs, over 300,000 base pairs, 364 exons, so it's made from little repeating units, and these units are broken up into bits of extra pages in the cookbook of the DNA, and these pages can be reassembled by putting them together in different ways. So you take a cookbook, you take different recipes, and you put them together, and you get a different menu. And these different menus in titin are called splice variants, and essentially they come in 2 different types of flavors, big and very big. But the important thing to realize is that these big things are made from repetitive units, and these repetitive units are quite small. They're actually only 100 amino acids, whereas the entire titin molecule is over 34,000 amino acids. So these repetitive units, this is terribly complicated, but it's the way it is, form little building blocks. They're a bit like Lego bricks, and these Lego bricks can stick together to other things and thereby assemble the sarcomere. And you could just think of it of Lego bricks, and titin then is just big, modular, multifunctional, dynamic, and elastic. And these are really the key features of titin. But, of course, something that is so very big can also fail, as the Titanic illustrates rather well. And over the last years, since about 2002, when our colleague Bjarne Ut in Helsinki identified the first titinopathy, a genetic disease caused by mutations in titin called tibial muscular dystrophy, very rare, quite endemic in the northern parts of Finland. There has been an absolute avalanche of mutations identified in the titin gene linked to myopathies, and these are just a few of them. Heinz has already mentioned some of them, and central nuclear myopathies are one of those that are now really transpiring to be very much more common than we thought. There's also very confusing things, because, for instance, this paper in New England Journal of Medicine from a few years back reported that truncating mutations, so mutations which make the titin molecule shorter than it should be, are very frequently associated with a cardiomyopathy, whereas these patients were not reported to have skeletal myopathies. This is very surprising because the sarcomeres in skeletal and cardiac muscle contain pretty much, for some little changes, the same type of titin, and most of these truncations are in the type of titin that is common to both skeletal and heart muscle. And what is even more confusing is that about 3% of the control population also has these truncating mutations. And if we look in what is called the 1,000 genomes, which is essentially just sequencing lots and lots and lots of genomes, then we find that actually the picture is even more confusing. There are tens of thousands of genetic variations in titin. Over 6,000, that was from May 2016, and I've just capitulated since updating this slide. Over 6,000 stop-gain mutations and over 90,000 missense mutations, which are mistakes replacing one amino acid with another amino acid. So this is a real problem. So what does this tell us? Well, really what we look at is a glimpse of human evolution, changes that occur over time and in different ethnicities and in different people that could be good, bad or ugly, and that reminds you of this. But how do we tell these guys apart? And that is the real problem. So if you've got this gigantic gene titin with mutations scattered from one end over the other, the real challenge is to tell the good and the bad and the ugly apart. And I'm going to illustrate that, how we learn how to do that with a few examples. So this is a pediatric patient with a very severe skeletal and cardiac myopathy that was identified by Carsten Bönnemann, actually, and we analyzed it together with Anna Ferrero in Paris and Carsten, and that's published work. And this patient carries two mutations in titin, a premature stop codon, so a truncation, and a missense mutation, an amino acid exchange, and at the very C-terminal end in the kinase domain that titin also has. And as you can see, both parents are healthy, also the carrier of the truncating mutation, and the patient has this severe affectation. And interesting enough, this missense mutation was just recently identified by Anna in another patient inherited with another truncating mutation. And this particular other patient has no skeletal myopathy because the truncation is in a different exon. So we get better also at predicting the impact. But we were quite lucky at the time because we had very detailed molecular information, which looks very complicated, including the atomic structure of the mutated domain. So we could predict what this thing does. It looks very, very destabilizing. And when we measured how readily by heating up the protein we can destroy it. All of you probably destroy proteins by heating them up, by cooking eggs or boiling stuff. All of that cooks proteins and destroys them, and we can do that more sophisticated in the lab by monitoring the stability over various temperatures and then seeing when the protein gets destroyed. And these melting curves here tell us how stable a protein is, and this protein is seriously destabilized. So we've looked over very many mutations over the last years with Heinz and other colleagues. No, you don't need new glasses. You're not supposed to read all of those. We've just looked at really many mutations. And the common thing that we see now is that in the centronuclear myopathies, there can be homozygous truncating mutations or heterozygous mutations with truncations and missense, and all of them cause the same kind of clinical picture. But with the amino acid mutations, it's really difficult to predict what they do. And the computational models, if you go into online tools for mutation impact assessments, they're complete rubbish. One of them is called iTasser. It should actually be called iTosser. It is really hopeless, and we've therefore decided that what we really need is an experimental approach. So we begin to solve the atomic structures where we can really see individual atoms of these domains. We can then see what happens if these mutate, and we can measure the impact of the mutation by doing this cook your egg and see when it melts approach. And we see that these mutations are seriously destabilizing. So the domain gets destabilized at much lower temperatures, and these are not body temperatures, very clearly. None of us take our baths at 64 degrees. But what it tells you is that the protein is destabilized. And this is something you might argue is just any mutation could do that. But in fact, when you look at mutations that are very common in the general population, so any one of you might have one of those, they're not destabilizing. So destabilization seems to be a feature maybe of titinopathy causing variants in titin. And to assess this more precisely, we've developed a web-based tool called TitinDB, which you can access on the internet. You can look up every mutation in titin, even the ones. That's actually my reminder to shut up now. So this web-based tool helps to assess bioinformatically the impact of mutations by using every existing or modeled structure of titin, and you can plug in your mutation and see what it does. And actually, it is pretty good. So every time you get a value called MCSM that is negative, it's likely to be pathogenic, but very clearly not always, because some of these neutral variations can also have negative values. But what is really predictive is if the domain is additionally thermally destabilized, then it is pathogenic. So this is common to other protein mutations causing myopathies. All of these mutations in different genes here cause different types of myopathies. So destabilization and protein misfolding could be what probably underpins the titinopathies as well. And that is similar to other diseases with toxic misfolded proteins, like neurodegenerative diseases, like Alzheimer's. So possibly we can learn from some of these non-muscle diseases something about potential therapeutic approaches to muscle diseases. And with that, I'm finished. I just wanted to remind you that titin is this gigantic protein that is required for assembling sarcomeres, very common mutations, very difficult to assess them because they can be recessive or dominant, and we really need to understand better the impact before eventually we can hopefully develop some therapies. So this is our team at London. We've got many collaborators all over the world, including our clinical collaborators in London and in other places, and we're very grateful for our funders, including the European Union and the Mytubular Trust, who funded some aspects of this work. So thanks very much.